Me too. Yeah. No, can I get annoyed when I get you know? I've been there. Oh, it's the bank. Hello, everyone. Hello, we're getting started. It gives me enormous pleasure to introduce today's speaker, Dr. Ann Yoder. Ann comes to us from Duke University, where she is the Braxton Craven Professor of Evolutionary Biology, as well as having been the long-serving director of the world-famous Duke Lemur Center. As we will see in today's talk, Ann is the world's leading authority on the evolution of the fauna of Madagascar, a topic she has been studying since she was an undergraduate. She received her PhD in biology from Duke University, where she worked with Kathleen Smith and Matt Cartmill, two names will be familiar to some of you, followed by a postdoc at Harvard with Mary Ellen Ruffalo. Her first faculty position was at Northwestern University before she moved to Yale University, where she was appointed Associate Curator of Mammals at the storied Peabody Museum of Natural History, as well as being Associate Professor in the Department of Ecology and Evolution. Shortly after receiving tenure at Yale, she received an offer she could not refuse to become director of the Duke Lemur Center and professor of biology at Duke, where she has been ever since. As we'll see in today's talk, Ann's research has been wide-ranging, studying the evolution, biogeography, and ecology of Madagascar's diverse fauna and to some extent flora, including studies not only on the island's famous primates, but many other elements of their unique biota, including bats, carnivores, lizards, frogs, and plants. As well as these studies, Ann's research is widely cited for its far-reaching implications for evolutionary biology, including demonstrating how rapid speciation can occur and the extent to which highly diverse lineages can originate from a single ancestor. Yoder has also produced influential work on phylogenetic methods. Her study of the incongruence length difference test demonstrated how this widely used method in phylogenetic reconstruction can introduce bias in estimating species relationships. And her research on the use of local molecular clocks in estimating species divergence times is widely recognized, and we'll have to turn to the page to see what it's widely recognized as contributing to a fundamental shift in how researchers employ molecular clocks in estimating species divergence times. Not surprisingly, based on these accomplishments, Ann has received many honors and accolades, including election as president of the Society of Systematic Biologists, the leading organization in this field, as well as being a recipient of fellowships from both the John Simon Guggenheim Memorial Foundation and the Alexander von Humboldt Foundation. Most recently, she was elected last year as a fellow of the American Academy of Arts and Sciences. Ann's talk today, as you can see, is Genomes as Metronomes, Madagascar's past, present, and future. Yeah, it's a pleasure to have you visit with us today.
Can everybody hear me okay? Okay, that was an applause-worthy introduction. Thank you, Jonathan. That was really nice. So I start, of course, by thanking all of you for your hospitality and for having me here. I've had an amazing day. Eric, I would not have lived without this. I just want you to know, it's like I've gotten about that much sweat. Yeah. So, and Jonathan, I just want to thank you very sincerely for your collegiality and for sharing your enthusiasm for this place, and I'm just delighted to be here. So thank you very much. So Jonathan gave you my pedigree, which, excuse me, hilarious that you brought up the ILD test, because this is, I won't even tell you the story, but it was a test that people were using to determine whether to combine different data sets. And we, had a decisive empirical result that showed, no, it was misleading. And so we wrote a paper, it was published in Systematic Biology, and it cited like crazy. I mean, it was like really a surprise. And somebody recently said to me, you just killed that test. I was like, I didn't mean to. So anyway, I'm going to talk to you about Madagascar. And this talk is far-ranging, and it starts with my dissertation work and it ends with work that I've done recently. So I'm, just to forewarn, it's going to be sort of skating over the surface of a lot of things that have a lot of complexity underneath, and I'll try to, do it efficiently and give the complexity where it's needed. Okay, and Living Earth Collaborative, Department of Biology, thank you very much. Okay, so I have worked across different timescales in studying Madagascar, and it's important because we want to know why it is the place that it is and how it came to be, and we want to know what's been going on, and we want to try to guess what might be happening. So I have worked in historical biogeography, and I've also worked in phylogeography, and those are two interrelated fields, but they do differ in this way, I think. So with historical biogeography, I think we're talking about deeper time scales and larger spatial scales. So, global, essentially. With phylogeography, we're looking at asking questions about more recent time scales, and we're typically looking more locally. So like biogeography is understanding Madagascar's place in the world, in my view, and phylogeography is understanding what's been going on in Madagascar since it came to be. So phylogeography, I love. It is the most interdisciplinary, I would say, kind of science that you could imagine. This is just, was off the top of my head, all the different types of tools and techniques that I have used to apply to phylogeographic questions in Madagascar. And so I'll be touching on pretty much all of these concepts during the talk. So the talk outline, to start with, we will, in the context of historical biogeography, we'll talk about the geology and history of Madagascar. Then we'll move into sort of historical biogeography of lemurs and other endemic terrestrial mammals. So this is where a lot of my early work focused, how, what, when. Then some, passing thoughts on divergence time estimation, which certainly I've given 3-hour lectures on divergence time estimation. So, you'll get 30 seconds of it. Then we'll talk about some of these phylogeographic models in Madagascar and how, and this was my insight last night as I was sort of fiddling with this talk, is the mouse lemurs, which I was going to talk about, but I'm not really going to talk about so much given the circumstances. But I realized that one of the fascinations that I have for mouse lemurs is that they are kind of this organismal metronome. So they're sort of keeping time ecologically and environmentally in Madagascar. So that's what drew me to them initially. And then we'll land on the future and climate change and some of the things that we're also concerned about right now. Okay, so first to get oriented, here we are in North America, and way around the globe, we find Madagascar. So it is the world's 4th largest island. It's off the coast of East Africa, right across from Mozambique. And it is a very large island. It's about 1,000 kilometers north-south and about 300 kilometers east-west. And, or is that miles? Yeah, I think that's miles actually. So it's a sort of a mini continent. And there's actually a quite well-known book called The Eighth Continent about Madagascar. And certainly Malagasy people, if you tell them that they live on an island, they'll say, no, we do not. We live on a continent. So it's a real thing. So I'm fascinated by this particular island. So there's a field of island biogeography. I hope I've contributed to that field, but that is not what's driven my research, it is really an obsession and a fascination with Madagascar specifically. And so as part of that fascination, I did a sort of a back of it, I mean, a truly back of the envelope calculation to estimate Madagascar's land surface area relative to the rest of the world. And it turns out that it's about 0.01% of the Earth's land surface area. So that is like not much. Yet, if you look at the fauna, at least of Madagascar, and specifically the primates, you find that almost 20% of primate diversity is found on this tiny speck of land on the planet. And so this was a pattern that I came to understand very early in my career. And when I was working as a technician at the Smithsonian, actually between college and graduate school, and it just, I don't know, it took a hold of me and it's never let go. So this was the driving question. Why so much diversity? Why Madagascar? And also, it's been known for a long time, and it's certainly only an accelerating phenomenon, that lemurs and pretty much all of the endemic flora and fauna in Madagascar are critically threatened with impending extinction. And the IUCN designated lemurs as the most threatened mammals on Earth. I'm not entirely sure that's correct, but it's close enough. And what you see here is a shipak with a recently deforested area nearby. So let's talk about the geology of Madagascar. So I'm going to walk out a ways. So this is a time ticker, and we started at 200 million years ago. This is the original continental landmass, Pangaea. And what we're doing is, if you look at the ticker up there, we're moving closer to the present. So what you start to see is the separation of Laurasia in the north and Gondwana in the south. And almost exactly at that same moment, here's Madagascar. It starts to separate from Africa. So to get you oriented, here's Africa, there's South America, here's Antarctica, India, Australia, up there is Asia, North America. And so what happens then about 130 million years ago is Madagascar separates from Africa, but it stays connected to India for about 40 million years. So this was, paleogeologists call this the Indo-Madagascar continent. And it stayed connected. So we're just going to hang out for a minute here. We'll get to 88, I've done this a few times. We get to 88 million years, and what you see is India then starts to pull away from Madagascar. And the next cool thing to see, I mean, you can watch your favorite parts of it if you want to, but I ask you to keep your eyes here. What happens is India then goes over a volcanic hotspot, sort of scoots in geological time, you're about scoot, and then collides with Asia, builds the Himalayas, all of that. And this is why it's called the Indian Ocean, because India did even pass right through it. So, but the point of all of this is to say that Madagascar has been all by its lonesome out in the middle of the water for a very long time, and separate from India for 88 million years. Okay, and everybody usually says, where did you get that animation? Chris Scotis at Arizona State, the Paleo Map Project. I encourage all of you to surf his website because it is a fantastic tool for teaching purposes. If you want any sort of geological information and maps, it's all there. So shout out to Chris Scotis. Okay, so the questions that motivated my early research and specifically my lemur fascination was how many times, when, and by what mechanisms did terrestrial mammals / lemurs colonize Madagascar? So those are the questions that drove my dissertation work and my postdoctoral work, and specifically focused on the lemurs. So I'm again just surfing right over this stuff. So this one slide is both my dissertation and my postdoc work. And so the culmination of it all was a publication in PNAS, Ancient Single Origin for Malagasy Primates, with my postdoc mentor and my graduate mentors as co-authors. And what we determined was that all living lemurs are a clade. and their closest relatives are their Afro-Asian lorises. And, it give you a lot of biology there. But so the parsimonious explanation then is a single origin in Madagascar. But then the question becomes when, because if it's ancient, really ancient, perhaps lemurs were already there and they just drifted away from Africa, and and India left them in the dust or whatever. So we need to know how old they are. Well, this was really early day. I was so obsessed with divergence time estimation back in these days before it was really even a field. And this is, and I'll come back to this in a minute, but what we did is we estimated around 55 million years for the basal node of the lemurs. So what does that imply? Well, before I get to what does that imply, one thing that when people are thinking about lemurs, like the average person, even the average scientist, you're just thinking about the living radiation. In fact, there was an entire megafaunal lemur component to primates in Madagascar. And the largest one, Archaeoindris, was about the size of a female gorilla. So can you even imagine what that would be like to encounter one of these things? And there were something like 16 different species of very, very different morphologies and no doubt ecologies. So I was obsessed, and it's like I have serial obsessions. So this was an obsession at a time that I wanted to know where they plugged in to the living lemur phylogeny. And the short answer is, as the title implies, ancient DNA from giant extinct lemurs confirms single origin of Malagasy primates. So what we see is that the very funky and interesting eye is the basal lineage, and these giant guys that are now extinct are actually nested in the rest of the clade. So this demonstrates that they, all of them, the living and the subfossil, were a single clade. And this, just to shout out to my collaborators on this, is Tom Delapousse and Praveen Corinth were postdocs who led a lot of this work. And this was done in collaboration with Tom Parsons, who I was telling Jonathan about, I believe, or somebody, who was the director of the International Commission on Missing Persons for a while and worked at the Armed Forces DNA Identification Lab. So I've been working with some really hotshot forensic scientists, and Tom being one of them. So thanks to Tom. Okay, but now to the point. So if lemurs did start to radiate in Madagascar, let's assume, at about 55 million years ago, this is what things would have looked like. So Madagascar would have already been entirely water surrounded. It would have been an island, and as it has remained. So then the question becomes, how on earth did they get there? If they're just, if Madagascar is sitting out in the middle of the Indian Ocean, how in the world are lemurs to have arrived there? Hold that thought. There are 4 groups of endemic terrestrial mammals in Madagascar. There are the lemurs, the primates, there are the carnivorans, there are rodents, and then there are these very funky Afrotherians called tenrecs. So, at the scientific, so this was my some assistant professor research. So I became interested in the carnivora, and I was collaborating with John Flynn, who was at the Field Museum. And so we decided to do some phylogenetics and divergence time estimation, looking at the carnivora to see what their patterns look like. And one reason that it was timely was because in 1997, a paper was published. I think it was in Evolution. I can't recall exactly what journal right now. It's been a while. That posited that there was an island chain that existed between Africa and Madagascar, and that it was, had very specific sort of time frame of 45 to 26 million years ago. So we were thinking, well, okay, the lemurs are too old, right? We, by our estimation, to have benefited from such a land bridge. How about the carnivores? Let's see what happens with them. And what we found, in fact, was the carnivora were much, much more recent than the lemurs. And the kind of amazing thing, I mean, you would think stochastically they would maybe fall into this putative land bridge time window anyway, but instead they're way too young to have benefited from this hypothetical island chain. So, this is my one and only Nature paper, and, people were pretty enthusiastic about that. And it turns out, and, just to digress for a bit, that we discovered that the Malagasy carnivora are actually derived mongooses. So it was like some kind of little mongoose got over to Madagascar and then turned into the, several crazy forms, including the Fusa, the notorious Fusa. So that's a derived mongoose that you're looking at. So again, to mention, there are rodents and there are these tenrecs. And cool thing about tenrecs, so this is one of the tenrecs that looks like a little hedgehog. This creature is much more closely related to an elephant than it is to a hedgehog, which I just, that's a little mind blowing. And I have a friend who said this one looks just like James Brown. So I always love that. So anyway, so this was very much in the water at the time, in sort of early 2000s, early aughts, as they say. And this Miguel Vences, I'm not part of this paper, but Miguel Vences, who's a colleague of mine and collaborator, who is very interested in these same questions. They published a paper where they looked at all 4 groups and estimated the phylogenies and the divergence ages. And they also found that they're scattered all over the place, which implies at least one thing, that it wasn't a single event that explains the origin or the presence of these groups in Madagascar. And this is an old slide, actually. I mean, there are many more studies that have been published since. See that, like, the most recent publication date is 2005. But it was, there was sort of a real heyday in the early aughts, late 90s, where people were just saying, well, how about this group? How about this group? And again and again, the pattern just repeats that it's single origins, and, they get to Madagascar once and they, do stuff. So, except for chameleons, and that's a whole other story that I can tell people who are interested over a glass of wine later on. And that's not my work either. Okay, so back to this divergence time question. So I mentioned to you that we got, this sort of approximately 55 million year ago date using really primitive methods, I confess readily, and with only 1,140 base pairs of mitochondrial DNA. Well, so fast forward to 2018, and this is, as Jonathan mentioned, I've been doing some divergence time work with Yang, who's at University College London. His former postdoc, Mario Das Reis, sort of led the charge with a much more recent attempt at, putting ages on dates of lots of primates, in fact. And we had 3.4 million aligned base pairs. So we're talking genome scale data. And look at that, the age is about the same, which I find reassuring and interesting. There's a manuscript in prep that I just got a abstract for, and I'm a co-author on here, which is again genome scale data, but with this very, very old date, and I'm a little worried about it. So I don't think it's right. Sorry, whoever, Katie, I realize this is going out to the world. Okay, so just a moment to talk about divergence time estimation. So the sort of state of the art now is to use genome scale data, and typically to use concatenated data, meaning that genes are just kind of treated as one long one long gene, basically. So whatever, if you've got a chromosome or you've got exons or what have you, and you treat them all the same, and then you apply models, and you typically test a phylogenetic hypothesis with likelihood methods, and this is how you determine best dates. But you have to calibrate your phylogeny with a date somehow, because it time and rate are confounded. So if you don't know rate, you need to know time. If you don't know time, you need to know rate. And if you don't know either, then you can't do anything, because you don't know if a branch is long because of a high rate or a long time. So it's super important in this case, that you have a fossil to calibrate the tree. Well, we have 0 fossils in Madagascar, and this is a phenomenon that I don't entirely understand, but every geologist I've talked to just says, well, we just don't have the exposures for the terrestrial record post-Cretaceous tertiary boundary. So we have some very recent subfossils like the giant lemurs I was talking about, but they're quite recent. They're contemporaries really of the living lemurs. So I just, to emphasize the point, no terrestrial fossils in Madagascar to calibrate our trees. And that's something that's always worried me a great deal. And I'll come back to that in a minute. Okay, so going back to the sort of biogeographic questions of how on earth did lemurs and these other mammals get to Madagascar, we sort of have 3 choices. We have the vicariate model that I talked about where they were just there and they drifted away. There's the land bridge, and we've already talked about the land bridge, the McCall hypothesis or island hopping. And then we have the sweepstakes hypothesis that George Gaylord Simpson made such a fuss about. And which one is it? So I think we've ruled out vicariance from the beginning because, Madagascar is already way out there in the ocean before eutherian mammals or, placental mammals really evolved. The land bridge, I think we dispensed with that with the comparison with the carnivora and also the Poe et al. Paper. So we're left with sweepstakes, and that's just what we're left with. So, so this, I love this drawing. This is a drawing by my colleague and friend, Stephen Nash, who does incredibly beautiful artwork. And he just sort of doodled this, and I use it shamelessly. Okay, so that's hard to get your head around, isn't it? That a group of mammals of any type, primates, rodents, whatever, could drift across this vast oceanic barrier. It's about a 300-mile wide channel, and it's very, very deep. It's the separation between 2 plates, so it's not that sea levels really could have made a difference. So how does that happen? How would they do that? And this is interesting work. I have to shout out to my colleague Dave Krause, who is a paleontologist and a very good geologist as well. And he has done a lot of sort of modeling based on empirical data on observed storm tracks that we see now. And so this is like taken from whenever the cyclone tracks Madagascar from the World Bank. This is the now time. And what happens is these cyclones come across the Indian Ocean from the east to the west, and then they bounce off the thermal inertia of Africa, and they can come back across Madagascar. So Madagascar really gets hammered by these cyclones. But the point is, it's this sort of sweeping of, severe winds and currents. And, so this is a potential mechanism. And Dave has a beautiful book chapter in which he talks about this, contemporary phenomenon. And you can see giant floating islands out in the Mozambique Channel that have been just pulled away from riverbanks. And so it's a mechanism. It's a today mechanism, and it's always a big, dangerous thing to say, well, it happens today, so it must have happened 55 million years ago. Well, that's a stretch. So, but it's it confers some plausibility. There's Madagascar. Okay, so this was a paper published by Ali and Hubert in Nature in 2010, and Henry G. was the editor in charge of this manuscript, and he sent me he asked me to review it, and I could almost see the smile on his face in the email where he asked me to review it. So Ali and Hubert are computer scientists and sort of geospatial modelers, and what they did was they simulated these paleo conditions across the globe, and with supercomputers at MIT and so on, because one of the complaints of the sort of anti-dispersal camp, which there is a very thriving one, is that the current ocean patterns are from the east to the west, so they would not be favorable for rafting from Madagascar to Africa. But by the modeling of Ollie and Huber, what they found was that, in fact, at this time, that, this window of time where the mammals or the lemurs and the rodents and the chinrex and the carnivorans might have rafted, the currents were actually moving west to east. So would have been favorable for a sort of sweepstakes dispersal. And this, I was not going to read this whole thing, but this was personal communication with Jason Ali. And he was explaining that, in fact, under their models, there would have been these superstorms about every, every century or so during this period, where the transport time from Africa to Madagascar would have actually been about 25 to 30 days. That's still a long time to go without food and water if you're on this raft. So it doesn't explain it away. So, but it does, again, bring up some plausibility to this model. But, and then Dave Krause wrote a very nice review of, all of the accumulating literature. And I think this was actually an accompaniment to the Ali and Huber paper. This, it tells the story of lemurs coming first, and then the chinrex, and then the rodents and carnivorans a little bit later. And actually, I had a deep discussion with some graduate students at UBC about, why lemurs and all these creatures have speciated so broadly and into all these different niches. And I'm thinking lemurs would, what was it like there when they arrived? Were there even any predators? That's an interesting question. So again, to talk about plausibility of this dispersal, there's actually this idea that maybe these various groups had the capacity for torpor or hibernation. And that holds some water. Again, it's there's a lot of hand waving going on here. There's a lot of storytelling, but in turn, it turns out that at least in the case of lemurs, there might be some weight to this. So can anybody see the mammal in this photograph? I will help you out. Okay, so this is work that my colleague Marina Blanco, that you'll hear me mention throughout the talk. She is interested in hibernation in the dwarf lemurs, and they are the only hibernating primates in the entire primate clade. And so she's done some just amazing work. And this is, I'm about to show you a video that she filmed, and there's an audio to it, but it's only going to come out of my computer. So just, tune your ears, everybody. Quiet.
Rehefa mitady hoe fa betsabetsaka ny hanina dia somary tsindrio lery manala ny hanina. Dia tonga dia mitsoka. Voilà. Mbola matory.
So this is a dwarf lemur that's hibernating, and the way she knew where to find it is she radio calls them in their active season, and then comes back the next year and goes out with her wand to find them. So I just, this is he looks like, really? I was asleep, thank you. Anyway, so this is all just to say it's still a very hard model to get your head around, and it's very uncertain, but I don't know of a better alternative. So we're going to rest there for a moment, and let's go back to the question of what Madagascar would have looked like to lemurs when they arrived. And so this is a paper again by Dave Krause, whose work I admire very much. So they've done a lot of the paleo work right leading up to the Chicxulub event. And he says our recent fossil discoveries indicate that ancestral stocks of Madagascar, overwhelmingly endemic modern vertebrate fauna, arrived on the island in post-Mesozoic times. So this would have been after the big extinction event at the KT. So that would imply that Madagascar also suffered, even though Chicxulub hit, in the Yucatan Peninsula, and it's often thought of as a sort of a northern hemisphere event that might have had a major impact, as it were, in Madagascar. And in fact, if you look at the pre-KT vertebrate fauna from the fossil record, this is the kind of thing you see. You see dinosaurs and you see these funky multi-sperculate and Gondwanathere. So they were mammals, but they're not placental mammals. They're just completely, they're all extinct. And then I couldn't resist grabbing this one off of the worldwide interweb. This giant frog eating a dinosaur or something. And then if we look at what today's vertebrate fauna looks like, it's a very, very different fauna. So this is an elephant bird for anybody who's never been exposed to this, the largest bird that we know of to ever have lived. A ratite and extinct now, and chameleons and so on. So this going back to this arrival time of these different groups of mammals that we have now implies that if lemurs were the first arrivers, that it would have been this wide open world where they had no real competitors, they had no predators. And so this might be why we have such funky looking lemurs, and tiny ones and huge ones, and doing all different sorts of things. So that's the way I like to think of the historical biogeography of Madagascar. Now we're going to move forward a bit in time. And the other aspect, or another aspect of Madagascar, and the part that has me by the throat right now, is that it has sort of been thought of as a speciation laboratory. I've told you that these different groups arrive, and there tends to be single colonizations. And in almost all cases, once they arrive, they do weird things. They turn into, you have a primate that acts like a woodpecker, and you have a, elephant relative that looks like a hedgehog, and it just goes on and on and on. So they definitely do interesting things when they get there. So let's go back to mechanisms, potentially. And so again, in the divergence time estimation realm, we've estimated now, we sort of settled on around 60 million years for the origin of the lemur clade. And one of the weird things is, this is a sort of a sidebar, is there's this long gap in the tree before you get the next big radiation, which is about 41 million years ago-ish. And so what's that about? I don't know. I have hypotheses, but again, that would be dinner conversation or cocktail conversation. But my point is trying to be that I'm getting closer to the tips now in the tree and trying to know what's going on up sort of in the top parts of the tree. So again, emphasizing no fossil record. What do we do about that? So this is work that I've done quite recently, and again, in collaboration with Mario Das Reis, and we're trying to develop methods where we can do fossil-free divergence time estimation. And the theory goes that if we can measure the de novo mutation rate, so these are the mutations that occur in the germline in every generational transmission, that are the engine of life, literally. Without these mutations, the engine of evolution would run out of fuel. There'd be nothing to work with. So there is this rate. There are these de novo mutations that arise, and the theory goes that you can use that mutation rate, and you can calibrate it with the generation time of the organism that you're studying. So that in that way, you can estimate an age without the need for fossils. So there's a lot of complications to this, and the questions about whether it's appropriately used at the tips but not the base of the tree and vice versa. It's very complicated. We actually sort of worked through a lot of this in a review paper, and this is what I do whenever I confront something new scientifically that I'm obsessed with and really want to know more about. I say, time to write a review paper. So that's what we did. I was asked to write a review paper on conservation genetics, and I said, I'll write a review paper, but it's going to be a little different. So this is just in case you're interested. I invite you to have a look at this paper, because we talk about the issues of concatenation and fossil calibrations versus these kind of fossil-free estimators. So going forward in the talk, though, this is the one reason I'm putting this here sort of abruptly, is that going forward in the talk, when I'm talking about some of these more recent events and the ages that we are ascribing to different events, we're using this fossil-free method. So it is quite different from the methods that have been used to, say, estimate the age of the lemur clade. Okay, so again, the question of like Madagascar as a speciation laboratory, why is that? Why is it? And there's time, there's space. And one thing, another aspect of it is that Madagascar is environmentally extremely heterogeneous for such a relatively small landmass. And anybody who's been to Madagascar knows that you can, drive 60 kilometers and start one world and end up in another one. Or you can turn a corner and you're in a completely different climate and vegetation type. So there's been a lot of empirical work and theoretical work around this idea of environmental heterogeneity, sort of as the driver of speciation, the engine of speciation in Madagascar. So we're trying to look at that empirically. Okay, so another thing to understand about Madagascar. So this is an animation that my former postdoc Jason Brown made for me. This is, we're looking at Madagascar from the south end up towards the north, and we're going to tilt it on its side a little bit and look at it. And you can see that it has rainforest on the east and it's dry on the west. And the east is very sharp. It's they call it the eastern escarpment. And if you think back on the animation that you saw, this is where India used to be. So this has set up the climate of Madagascar in many ways, in the sense that, because of this escarpment, the moisture comes off of the Indian Ocean, it hits this abrupt altitude and it dumps the water. So that's why we have this very wet east and very dry west. And so that really structures a lot of our thinking about how biodiversity is distributed in Madagascar, and how we think about the evolution of these various groups. So in a cartoon form here, we have the rainforest on the east, dry deciduous forest on the west, and then we have this high plateau, which is largely grassland and with mosaic forests. So this gets into human impacts. So this is the hypothesis that we're now working under. I think this slide actually should come a little bit later, but I'll just go ahead and give it to you. So prior to human arrival, we are hypothesizing that the central highlands were woodlands, bushland, and open areas, and that fire from human activities dramatically changed the landscape, but only about 1,000 years ago. Humans are thought to have arrived in Madagascar about 2,000 years ago. So it's one of the most recently colonized land masses of any size on the planet. There is a lot of recent uncertainty about that age. Like some people have even said as old as 10,000 years ago, but certainly 10,000 is maximal age of human arrival. So that fire activity, slash and burn agriculture would have been a primary cause for the spread of secondary grasslands, and that forest fragments along streams may have been isolated far earlier than 1,000 years ago, and movement of animals from them may have occurred during times of warm cycles. So this is sort of a model for basic phylogeography, and also trying to ask the question about these central highland plateaus and are these grasslands anthropogenic or are they natural? So that's been a strong motivator for some of the recent work that we've been doing. And just to go back to this east-west division, so whichever group of organisms that you're thinking about, you would expect to see a quite distinct phylogenetic relationship separating Eastern populations from Western populations. But in fact, and this is where we're bringing in the mouse lemur, we are not seeing that at all. What we're, the early work, so this was some work that was published in 2000, and this is when I first got bitten by the mouse lemur bug, as it were. And we were describing new species and we were looking at phylogeographic patterns, and they were not holding up at all as we expected in this east-west pattern. It would look more like a north-south pattern. And we were seeing lots of species, and that's another talk that I can give another day. So different models have been proposed for, why not east-west, why this apparent north-south. There's a watershed model that actually I'll spend a little time with, because this was published in Science in 2006. And if you think about the geography of Madagascar with the highland plateau, the water runs down, and it's running down east-west all across the island. So there are no north-south river structures at all. So everything is east-west. So then if we're thinking about rivers as possible biogeographic barriers, this makes perfect sense then as to why you might see more north-south divisioning than east-west, as we saw. So we wanted to investigate this with a little more care. And here comes the mouse lemur or metronome. And again, another reason that I've been so interested in these animals, they have short lifespans, they have high reproductive output, they have short generation times, they live in all forested areas of Madagascar. And so hypothetically, they will closely track the environmental change through time. They will, as the forests expand and contract, so will the mouse lemurs. And, there are lots of them. And, we should be, and they, because of their very short generation times, we should be able to, capture fast moving events. And so, which we can observe with genetic or genomic data, and thus the genomic metronome analogy that came to me at midnight. So that's the mouse lemur model. And the kind of methods that we use, this is what, again, Jason Brown prepared this cool animation for me. So we can do phylogenetic analysis here. In this case, it's a lizard, a plurus. And then we can do spatial modeling to see, if we're asking, are these two clades different species? If we do the spatial, the ecological niche modeling, we see very clearly that the niche models are entirely different for these two clades, which gives us some rationale for calling them different species. So you can see I'm just kind of blurring all of these different things together: species delimitations, ecological niche modeling, phylogenetics, phylogeography, but it's all, it's all integrated to ask the same questions. All right, now I'm going to jump into an empirical study from the fairly recent past. My colleague Marina Blanco, who I mentioned earlier, who studies dwarf lemur hibernation, was in Madagascar, and I had funded her to be there. This was when she was still a postdoc, and she was collecting mouse lemur samples around the island for our, for me basically, because she's a dwarf lemur person. And there's a forest fragment called Ankafu Bay, and shout out to the Missouri Botanical Garden who curates this little forest. And it's just north of Tana. And she collected some mouse lemurs there. And she had even said, can I go to this place? It's only about an hour and a half from Tana. And I said, oh, sure, why not? And she brought them back. We did the genetics on, it was like 7 samples. And it was crazy. The amount of genetic diversity in that one little fragment was off the charts. I mean, it was just nuts. And so it was one of those moments where your head blown. So we ended up getting a PNAS paper out of this because we just started digging to find out what was going on. And and this is just a look at Ankafu Bay, and the size of it's just a tiny fragment. And there's a little mouse lemur, poor little guy. They all go free after they've been molested a bit. And so what does Ankafu Bay look like in context? We're zooming right in on it. And now we're going to pull back. This is always successful. So you pull back, and this is what the Central Plateau looks like. And that slide that I sort of laboriously went through about, human effects and the time and everything. This is what we're asking: is this an artifact of human activities, or does this at all resemble sort of, quote, natural conditions that were pre-human arrival? So And this project kind of started us on this particular track. I don't know what this, okay. So just to cut to the main chase of this study, so we used RAD-seq data, which are randomly distributed SNPs across the genome. It's amazing, fast, cheap, and easy, I say. And we used a program written by Graham Coop's group called SpaceMix, which what it does is it takes your genetic data, the little solid dots are the actual locality of the sample that you collected. And then these big sort of circles are the probable ancestral distribution of that genotype. So you can sort of map that onto the Earth and say things about the sort of geographic relationships of the present day and the past day. And so what we see, the two main points, the two take-homes in this diagram. So those blue circles, that's the mouse lemur species that we found in Akapu Bay, the little forest fragment. So this is Microcebus lehilatsara, which means good man in Malagasy, which is named for Steve Goodman. So it's a very clever name. It's my favorite mouse lemur name. So what you see is lehilatsara, it looks like they were, they've been sort of fragmented through time. And so this is, this is not recent. This is not in the last 1,000 or 10,000 years. This is, old time. And what you also see is that the closest sort of ancestral relative for this little dot over here on the East Coast is all the way over in the West. And, this is demonstrated phylogenetically and with other tests too. So it's saying two things at once. One is that the central highland plateau has probably been fragmented for a very long time, but in the South, there had to have been forest corridors across the island for these animals to, share these genetic profiles. So two things at once, saying fragmentation is natural, but also forest corridors were natural. So we're pursuing this even further, and this is a paper that was just published this past year in Molecular Ecology, led by my very recently former postdoc, George Tiley, who's marvelous. And this is sampling more broadly this species Lehilatsara to see, what broader geographic sampling than what we had in that earlier paper, what it has to say. And what it has to say, so this is either structure or admixture, I can't remember which plot. And this is like within species genetic diversity, and these are RAD-seq data, so they're pretty conserved. And what you see is like a lot of structure within this one species across the landscape. So again, it's backing up what we saw from that. It's just really, I love science. It's one of these things where you find some weird result that you just can't figure out, and you start really just obsessing about it, and you collect more and more data, and then you finally start to figure it out. So what we're figuring out is that indeed we hypothesize strongly that the Central Plateau was not continuously forested before humans arrived, that it had to have already been a patchwork, and that, perhaps human effects have further altered it, but that it was already pretty fragmented. And so this is a PSMC plot, which, I can explain over a glass of wine, but this is looking at whole genome sequences and basically looking at levels of heterozygosity. And so what you see is effective population size on the y-axis, and then time before present on the x-axis, with the corner being the origin being the now. So what this figure is showing you is that there, here's effective population size. It starts out at a fairly low level, but still probably healthy. And then there's a big kind of bloom in effective population size, and there's a dip here and another dip there. And what are these dips? Are they coincident with anything of interest? Well, the first dip is coincident with the last glacial maximum, suggesting perhaps that this was a Pleistocene climate cycle kind of phenomenon. But we then see that second dip, and that does coincide very closely with human arrival. So what it seems to suggest is that, Lehi was already in decline in numbers, but then humans have pushed that even further. So, and this is where we come to the now. Malagasy people need to eat. And one way that they do that is slash and burn agriculture. And it's not sustainable, and it's, damaging the environment. But again, people have got to eat. So it's a very complicated situation. We know from satellite and Bob Sussman, and this will be familiar to many of you here at Washington University. We look at, this is from satellite data. This is, I think this is an estimate that we're because we could do satellite, right? So this is an estimate of what the forest cover would look like in the east, but then satellite data show this, chewing away at the forest over time since human arrival. So we definitely know that deforestation is a serious threat to biodiversity in Madagascar, and we want to understand that and want to prepare for it, or address it. So just to, many of you will already know a lot of this about Madagascar. Humans arrived recently. The current population is about 28 million people. This is taken from the World Bank, so trust it. Population growth rate is about 2.68% per year. The per capita annual income is only $740. So this is, this is poverty. And it's, most of the environmental damage is subsistence needs, just eating and feeding your family. So again, very complicated. And we know that the planet is changing around us, that global climate change is, global weirding is my favorite term for it lately, because it's not just global warming, it's just global weirding. But in any event, Jason Brown, who I've mentioned several times already, who does this superb spatial modeler, I said to him when he was a postdoc with me, I said, what do you think is going to happen to lemurs in the face of climate change? So he said, well, let's find out. So we published this work in Ecology and Evolution in 2015. This is a very brief overview of what spatial or species distribution modeling looks like, which is basically, it's magic. You take locality information, you have climate data from, databases like WorldClim, and there are others, and then you do some nice math, and then you can project, ideal habitats. And we've already seen that, with the animation of the phylogeny on the landscape. So this is why I just have to shout out to Jason, because he just dug deep to get every locality date, every locality point that he could possibly find for lemurs. And so he dug into museum databases. He looked into primary literature, but this is a shout out to the IUCN and to Lemur News, because he went and he read every single article in Lemur News since it started its publication some 30 years ago, and collected the locality information from these, and this is like, if you know it, publish it, even if it's in a place like, that most people don't read, because someday you never know when it's going to be useful. So yeah, so these are out of order. So anyway, the idea is then to estimate ideal habitat in the future with these methods and all these data. And what we found, what Jason found, frankly, is, he identified areas of stability, areas of expansion, and areas of contraction. And then what he did was superimpose these different phenomena on top of each other and identified the places in Madagascar under these models. Again, it's modeling and future forecasting, but identify the parts of Madagascar that are going to be critical for lemur populations to exist and to move around. And you can see there's a red hot spot up in the northeast near the Massawa Peninsula. And so that what we landed on with the paper was that we need to focus on protecting these areas, first of all, but secondly, creating corridors so that lemurs and other flora and fauna can continue to move from one place to another as they need to do. To end, what we're working on now, mouse lemur speciation and some other things. One of the major goals of the project is to transfer knowledge and capability so that you can do in-country genetic analyses. And we are collaborating with Fidi Ross, as he goes, he has a wonderful Malagasy name that's hard to pronounce. But he has started a lab in Tana where they have -80 freezers and, you know, gel rigs and pipettes and, you know, all the things. So you can do some pretty nice genetic work in Tana, which is a big contribution, but it is in Tana. And so we're working with Fidi on our project. And one of the early stages of this work has been to build a sort of an Oxford nanopore mini PCR little mobile lab. And this is Marina Blanco, who you've heard mentioned several times, and her wife, Lydia Green, who is a postdoc with our project. And they held these workshops in Madagascar to talk about the, you know, not just the methods, but the questions and the, you know, why it's important. And as you can see, they're very well received and just a lot of enthusiasm. But the issue here is that we want to do something that is real, so that it's not just, you know, helicopter science that you come in and do stuff and go away. So that's what we're really thinking. We're having a hard think on that right now. Stay tuned. I don't know what the answer will be, but Oxford nanopore could be part of the solution. So with that, 1 minute over the time, I just want to thank all of my collaborators and my funders and field collaborators and wonderful people in my lab. And again, to thank you all for coming.
Mr. Rand would be happy to take some questions.
Yes.
Yeah, I wonder, is there any evidence is there evidence when you reconstruct your phylogenies of past hybridization or admixture among species?
Oh, that's a great question. Well, so we haven't looked at, my group has not really looked at that explicitly in any other group than the mouse lemurs. And we do see, so we use programs like BPP, Zihang Yang's program, and there's another program, GFOX, and there are multi-species coalescent, you know, gene tree, species tree types of approaches. And we can, you know, model ancient integration, but it in places where there's sympatric, it stops. Yeah, so this is part of the, would have been the mouse lemur talk, is getting into this issue of like reproductive isolation in sympatry, so that these, you know, crazy, they're like 25 described species at this point, and they all look to us exactly the same, and we don't know why they're speciating the way that they are. But yeah, so we're very interested in that, and we actually want to find hybridization, because that will allow us then to look for barrier loci in the genome that might be, you know, preventing gene flow or, you know, fusion of already diverged lineages. But I do know that another former postdoc of mine, Dave Weisrock, has a project where he's looking at that in the brown lemurs, and that's a perfect system for that, because their, you know, their geographic distribution is much easier to work with, and also they have phenotypically you can recognize them as the different subspecies, species, whatever, you know, diverging lineages, as I kept saying today, to avoid the S word. But yeah, there's definitely a lot to be done there, for sure. Thank you. Yeah. Somebody on Zoom, apparently.
Yes, we have a question from YouTube from Ari Miller, who says, fantastic talk. Thank you. 2 questions. How conserved are the pedigree-based mutation rate estimates across mouse lemurs? And 2, why might the II lineage be so species poor compared to the lemurs?
Oh boy, those are 2 very different questions. Well, the asking about how how fluid the rate is across mouse singer species. I wish I knew. The estimating those rates requires whole genome sequences of pedigrees. So you have mom, dad, and offspring, and you know the state of the art is to actually have a third generation so that you can test heritability patterns from the first transmission to the second, you know, with the idea that you observe a mutation you call de novo in the first passage, and then there should be about a 50% heritability of that mutation if it's a real biological mutation. So we only have one species in captivity that we can look at, and that's Microcebus murinus. So we have a rate estimated. It's a high rate. It's the highest rate for any primate, certainly, and that to me makes sense. I would love to know, but I'm not sure that we ever will by these methods, because it requires this very you know, close understanding of relationships and the whole genome sequencing. The I don't know. There was another, you know, named species called Daubentonia robusta, I believe, and it was exactly morphologically identical, but about twice as large. And so it was one of the sort of giant lemurs that went to extinction during this extinction window that wiped out, you know, all the large bodied ones. They're, you know, it's like monotypic lineages they're a puzzle in and of themselves. I tend to be drawn to the ones that are more diverse, you know, because I want to understand the diversity, but stasis is also interesting and needs an explanation. And first of all, the honest answer is I don't know. But eyes are super interesting in that as long as there are trees with, you know, grubs in them, it doesn't matter where you are. So they're pretty much island wide. They're not as tied to different forest types as other, you know, mammals are. So, you know, I basically cannot answer that question, but I can prattle on about it for a little bit. Thanks for the questions.
I want to follow up on mutation rate questions. So I believe like from a pedigree of 3 generations, you can estimate the mutation rate may be accurate for maybe 10 or 20 generations. But back to millions of
Oh, I know, yeah. Well, carry on, sorry.
It'll be like hundreds of generations, and a single error can get amplified pretty much. And how do you like manage the error exponentially grows without any fossil calibration?
Right. So the question is, just to repeat it as I understand it, is the question is, how about going back in time with these mutation rates, and what about the you know, biological flexibility of these rates, and how do we model for that? And doesn't it get more and more complicated the deeper in time you go, right? Is that the question? The answer is yes, of course, and that we discussed that in the paper that I highlighted, the Trends in Genetics review. And so our thinking is that it is a fairly good approach, I think, when, and this comes into the, it sounds like you certainly know what you're talking about in the sense of incomplete lineage sorting and, you know, recent speciations where there's a problem with these concatenated methods, because every gene you choose is probably going to have a different coalescent time. But once you get deep enough in the tree, they're all going to have reached reciprocal monophyly, each gene. So I think this is a better method for those more recent expansions like we're seeing in the mouse lemurs. But yes, it accumulates error and statistical error like crazy, I'm sure, the deeper you go in the tree. So it doesn't work all the way down, you know. I mean, it probably, I don't even know when it gives out, because, and also if you have a lot of rate variation, but this is true of the molecular clock generally, and that's a statistical approach, is you need to, you know, have the appropriate models. So I totally agree with you that it is not straightforward, but I do still argue that it's the best approach for these recent radiations when you don't, especially if you don't have fossils or some other temporal way to calibrate. Anybody else?
I have a comment on the overseas dispersal by the mammals and Larry Higney from the.
Oh, hilarious, yeah.
He published a general theory of mammalian diversification in islands like the Philippines, and his model has a very strong component of dispersal across the sea. And it doesn't happen on a scale of maybe a million years, but on a scale of 10 million years, getting a colonization is almost dependable. And so his model is very consistent with what you were arguing, and I think might be helpful in making that argument.
Yeah, no, and George Gaylord Simpson, I think, was the first one to say it. It was anything that isn't impossible becomes probable given enough time. And so that's kind of the argument. And Larry Haney, by the way, is a good friend, and I saw him just a couple of weeks ago when I was in Chicago. So yeah, Larry and I think very much alike. And thank you though for that comment.
So why don't we thank Ann again and retire next door?